Final talk is being given by Irina Ianculescu, who got her PhD from USC also in molecular biology, and she joined Pacific Meso Center a year and a half ago, or something like that, almost 2 years ago. And she's been working on the molecular changes in mesothelioma and the mechanisms that we can use in the future to tie in new treatments. So Irina is going to talk to us about those.
Can you guys hear me okay? I have issues with microphones. So my name is Irina. I'm a molecular biologist, and I've been at PMC for about 2 years now. And today I just kind of wanted to talk to you about genes and molecular changes that occur in mesothelioma. And I'm just going to give you sort of a little bit of background on it and tell you what we started to do in the lab and where we hope to go from here. And this is in no way meant to be a fully comprehensive talk about genes and every gene that has been found to be important for the development of mesothelioma. It's just sort of a brief outlook or a brief look at some of the more prominent research that has been done and what's out there. So what's important is for us to try to understand what the molecular mechanisms behind the molecular mechanisms behind the genetic and epigenetic changes that occur in the development of mesothelioma, and then how we could use this information to either find better treatments or be able to detect the disease earlier or at an earlier stage. And although a lot of work has been done in this area, progress is still lagging behind other more common cancers. So, before we talk about genes, I wanted to talk about cells and cancer cells versus normal cells. So what is the difference between a cancer cell and a normal cell? I know this might be kind of an obvious answer to some of you, but I just wanted to go over it really quick so that everybody's on the same page when we start out. So here we have a little diagram. We're looking at some normal cells that are shown in this little purple-pink color here with a tumor forming in the middle. That's colored, that's this blue color right here. So some of the characteristics of normal cells have this thing called contact inhibition, which means that normal cells will actually stop growing when they come into contact with the cell next to them. So there is a certain limit to the number of times that a normal cell will grow and divide. And this is represented by controlled growth. Normal cells also are specialized cells. They have particular functions that they perform. They exhibit a normal number of chromosomes. And finally, after a certain number of times that they grow and divide, they will die or undergo apoptosis. Cancer cells, on the other hand, lose most of these abilities, so they no longer exhibit contact inhibition, so they'll just kind of grow next to each other and on top of each other, and they don't really care that there's a cell next to them right there. They also have these very large nucleoli and these poorly defined boundaries. So whereas you can see normal cells are pretty regularly shaped and they kind of look normal, as you would guess, cancer cells have this very large nucleus and a very small amount of cytoplasm. So they experience uncontrolled growth, they lose any specialization that they had, they usually exhibit an abnormal number of chromosomes or just abnormal chromosomes in general, and they no longer undergo apoptosis, so they no longer die, they just kind of grow and divide and grow and divide. And some of the main differences comes down to basically their genetic makeup. So how do genes actually cause cancer? So it doesn't take just one mutation in a cell to give you cancer. It actually takes multiple mutations in a cell. A normal cell, in order for it to grow and divide, it needs to replicate its DNA, it needs to replicate its hereditary information. So when it does that, it grows and it divides into two separate daughter cells. DNA can become mutated by a variety of different factors, either by heredity, you can inherit a mutation from either of your parents. If you're exposed to radiation or chemicals, you can cause mutations in your DNA, or by spontaneous errors during DNA replication. So like I said, your DNA has to replicate in order to divide into 2 separate daughter cells, and sometimes errors occur. The cell has particular machinery that fixes these errors. The particular machinery comes from DNA repair genes. So DNA repair genes are really very important because they can go in and fix these mutations that occur in your DNA. The problem is, if there's a mutation in your DNA repair gene, you're obviously not going to fix any errors you have in replication, and this will lead to more and more mutations, and then you basically have a certain number of mutations that your normal cell has acquired, and it goes on to experience uncontrolled proliferation and growth and carcinogenesis. Another group of cells that can lead to cancer, or excuse me, another group of genes that can lead to cancer are called proto-oncogenes. So proto-oncogenes are normally are important for normal growth. If a proto-oncogene becomes mutated, it becomes an oncogene, and it goes through and it causes the cell to grow and divide uncontrollably, leading to tumor growth and formation. A third category of genes that's important are called tumor suppressor genes. Tumor suppressor genes are normally present in cells, and they act sort of as the brakes of the cell cycle. So as the cell is replicating, these tumor suppressor genes kind of do what they say. They kind of suppress the growth of tumors. So they're responsible for really controlling the growth and the cell growth and the cell death. So when these genes become mutated, that no longer occurs, and then again you get uncontrolled proliferation, growth, and carcinogenesis. So these are just some of the ways that mutations can cause cancer. Let's talk about what exactly a gene is. So a gene is basically just a short segment of DNA. You have many, many genes present in your cells. You have DNA present in your cells. DNA needs to be transcribed into this copy called RNA, because DNA cannot leave the cell nucleus, but RNA can. And it goes out of the cell nucleus, this RNA transcript, and it can make a protein. And a protein is sort of the physical manifestation of a gene. It's kind of the final product, if you will, of a gene. And today I'm going to talk to you about gene expression. And when I talk about gene expression, I'm really talking about RNA and levels of RNA. So it's really important that you understand that you have like one gene in your DNA, but you can have many, many, many copies of RNA. And that's how your gene is expressed. That's what we call gene expression. And so this is what we're going to be trying to measure, because we expect that there are different levels of RNA, different levels of gene expression that occur between tumor cells and normal cells. So in case this is a little fuzzy for you, I have a little analogy. So DNA is basically kind of like your instruction manual. So if you wanted to build an airplane, which would be your sort of final protein product, this would be your instruction manual, which, let's say you find it in a library. You can't take it out of the library. So what do? You go into the library and you write down on pieces of paper the instructions that you find in here. You can take those little pieces of paper outside the library, and you can go outside and build your little airplane, which is analogous to your protein product. So again, we're just looking at this RNA transcript here. So what can we do with genes? So like I mentioned, excuse me, we want to know what the difference is in gene expression. We want to know which genes are expressed by tumor cells. We want to know which genes are expressed by normal cells. Again, these types of genes should be different between the normal and the tumor. And then what can we do with this information? We can theoretically, hopefully, detect mesothelioma at an earlier time point. We may be able to determine specific treatment options based on the gene expression patterns, and we can also hopefully monitor response to treatment. So just by which genes are being expressed in your cells or by your tumor cells, you can monitor if the treatment is working or not. The question, of course, is which genes. So which genes are important in the development of mesothelioma? And this is basically what I feel like, because there are so many papers out there that name lots and lots of genes. And there is no one answer. There is no one clear gene that's really important for mesothelioma thus far. So you may have heard of BRCA1 for breast cancer. Mesothelioma, unfortunately, has no gene that is strictly associated with the development of this disease. There are a lot of candidate genes that are of potential interest that have been found to be either mutated in mesothelioma or differentially expressed between tumor and normal cells. However, for most of those, I should say, they also indicate a susceptibility to developing other cancers. So today, I'm just going to highlight sort of a few of the more important ones that are out there in the literature and the research. Alterations in meso are abundant. They kind of come in these 4 categories: chromosomal abnormalities, genetic alterations, which are clearly associated with oncogenesis, epigenetic alterations, which also have been shown to play a role in gene activity and tumorigenesis, and where our interest is changes in gene expression. So when I talk about chromosomal abnormalities, I'm talking about changes in chromosome number or structure. You have 46 chromosomes in your cells. Any change in the number or actual structure of these is associated with mesothelioma. Deletions, duplications, or translocations of chromosomal regions. So if you delete a part of your DNA or a part of your chromosome, if you duplicate a part of it, if one part of the chromosome moves from one location to another, that is also associated with development of mesothelioma. And in this disease, losses are more common than gains, meaning that in meso, people usually see deletions of chromosomes or deletions of pieces of chromosomes rather than duplications or translocations or anything like that. And there's something interesting called this EWSR1-YY1 gene fusion. It's actually a fusion of two separate pieces of genes. And this has been detected in mesothelioma. And what happens is this gene actually codes for an abnormal transcription factor that has a role possibly in the development of mesothelioma. So if you remember a gene, DNA makes RNA, makes protein. So if you have this sort of new novel gene fusion that doesn't normally occur and you get an abnormal protein, it can go on to do very detrimental things. How often this gene fusion is actually detected in mesothelioma is still the topic of active research, so that basically remains uncertain for the moment. Genetic alterations, you kind of heard about these 3 genes earlier today. CDKN2A, cyclin-dependent kinase 2A, is kind of one of the big ones. BAP1 and NF2. All of these 3 are tumor suppressor genes. They are all either inactivated or deleted in mesothelioma. So again, deletions and inactivations are far more common than any sort of gain-of-function mutations that occur in these. And any sort of activating mutations in oncogenes, as has been mentioned earlier today, are actually really rare in mesothelioma. Epigenetic alterations. So epigenetic might be kind of a new word for a lot of people. It's not as common, but it's definitely a popular area of research these days. When I talk about epigenetics, I'm talking about heritable changes to gene expression, not involving changes to the DNA sequence itself. So again, we're talking about changes to the number of transcripts, the number of molecules of RNA that we see versus the DNA sequence itself. And the way that occurs is through things like DNA methylation and histone modification. So DNA methylation is where the DNA itself acquires a methyl group. It doesn't actually change the sequence, but it just kind of silences it. And DNA methylation is known to downregulate the expression of a number of genes in mesothelioma. And what's interesting about DNA methylation is that there's a certain pattern of DNA methylation that occurs in mesothelioma versus normal cells. And then that pattern of methylation is actually different in mesothelioma than it is in other tumors, basically indicating that there's sort of like a signature methylation pattern that occurs for mesothelioma, which of course is of potential interest for further research. The problem with DNA methylation, however, is that it's kind of dependent on other factors such as age, ethnicity, subtype. So there's still a lot of work that needs to be done there. But really quickly, this is your DNA wrapped around histones. A gene is switched on because you can make RNA transcripts out of here. So when you methylate the DNA and when you modify the histones, you basically switch the gene off by compressing it, and you can no longer get your RNA transcripts. MicroRNAs. MicroRNAs are a hot topic these days because of their potential to serve as biomarkers in mesothelioma. They're short stretches of non-coding RNA that post-transcriptionally regulate gene expression. So by that I mean, if you remember, DNA makes RNA makes protein. These little pieces of RNA don't make any protein, but they do go on to have certain functions in gene expression that tend to silence genes. And also their expression levels tend to vary between normal and tumor. So for example, MIR31, microRNA 31, is frequently lost in mesothelioma. MicroRNA 29C has higher expression in epithelioid mesothelioma in patients with a good prognosis. So potentially, not only could you use MIR29C to detect mesothelioma, but you would theoretically be able to detect its subtype or its potential for being epithelioid versus sarcomatoid or biphasic. MIR15 and 16 is also downregulated in mesothelioma with respect to normal mesothelium. So like I said, our interest lies in gene expression, and excuse me, there have been quite a few array-based studies, array-based gene profiling studies looking for differences in gene expression between mesothelioma and normal mesothelium. And instead of going through every single gene that has been found, because I guarantee you there are a lot, I will tell you that genes involved in proper cell cycle regulation are one of the big groups that always come up in these gene expression arrays. Proper cell cycle regulation is obviously important for controlled cell growth and division. And just for your interest, genes like UBE1L, CCND2 are downregulated in mesothelioma. Checkpoint kinase 1, CCNB1, CDC2 are upregulated in mesothelioma, as are aurora kinases, which are important in the process of cell division. Because I work with an immunologist, we also have a huge interest in immune response genes, which we have seen genes such as CCL, CXCL, and this is supposed to say IL-4 receptor, and VEGF, which we all know to have enhanced expression in mesothelioma. So we wanted to look at gene expression in the lab with the available cell lines that we had, because when I started out, we didn't have access to actual tumor samples yet. So our goal was to develop this 3D tumor model to mimic mesothelioma ex vivo. So if you know about cell lines, cells, when they're grown in culture in the lab, they are grown as a flat 2D monolayer, which really isn't how they grow in the body. If you look at this picture of mesothelioma, you can see it has these little nodules here on the surface. And it has been shown by others before us that cells grown in this 3D spheroid structure, so these ball of cells are no longer grown as a monolayer. They're kind of growing together in this little ball that's not attached to anything. It's just floating in media. These 3D spheroids have been shown to more accurately mimic tumor biology in vitro, than cells grown as a monolayer. And this model, this 3D model, can then allow us to investigate the malignant cell behavior more accurately just by using these 3D spheroids. The advantages of using these 3D spheroids: they mimic cell-cell contact that's normally found in the tumor. Reproducibility is a big one. It's pretty easy to put these cells together and grow them as a little spheroid. It's much more, it's easier to manipulate the external environment. So like I said, they grow in a media environment. And as we all know, the tumor microenvironment and the tumor they interact together, and tumor development is very dependent on the composition of the tumor microenvironment. We are also very easily able to control the composition of multicellular spheroids. And because we can grow them in these little wells of a 96-well plate, so we can put one spheroid in each of these, we can basically test hundreds, if not thousands, of different conditions and treatment options, which is more feasible than using hundreds of thousands of these little guys that tend to bite, I've heard. So, one of the first things we did was we did our own Illumina microarray expression analysis. We wanted to show that there was a difference between cells that we grow in a monolayer versus cells that we grow as spheroid. This has been shown by other groups before us, but we wanted to do it using our cell lines to establish a baseline for our research interests. So H28 cells are an epithelioid mesothelioma cell line. MSTOs are biphasic mesothelioma, A549s are lung adenocarcinoma, and our MET5As are normal mesothelial cells. So we took all of these cell lines, we grew them both as a monolayer and as a spheroid. We extracted RNA because, again, we wanted to test for the level of transcripts. And we took that and we hybridized it all to a whole genome expression array, this Illumina expression array that contained over 47,000 unique genes found in the NCBI-RefSeq database. And what we found was that many of these genes are differentially expressed based on simple growth conditions. So we found lots of genes that were differentially expressed between the monolayer and the 3D spheroids. The good news is that, yes, we had a lot of candidate genes. We don't know what all of these genes do, but some could potentially just be used as a marker. This is some of the data that we got. So when we looked at spheroids versus monolayers for our 4 different cell lines, the red is upregulation of expression, which means that we had 3,024 genes that were more highly expressed in the spheroid versus the monolayer model. For this H28 cell line, we had 2,800 genes that were expressed less in the spheroid versus the monolayer model. I don't expect anybody to remember all of these numbers. My point is that there are many, many, many genes that are differentially expressed simply by growing them as a monolayer versus growing them as a 3D spheroid. And what's interesting about this is that it could, the difference in genes could explain why genes that have been found using the normal 2D monolayer cell model have failed to become clinically relevant, because potentially mesothelioma is acting more like this 3D model, and any genes that we find in this 3D model would be probably more clinically relevant. This is just to show you the overlap in the number of genes. So if you look at MET5As are our normal control cell line. Compared to MSTOs and H28s, you can see there's about 1,462 genes that are differentially expressed in meso versus normal when you compare the 2 different growth conditions. If you look at spheroids versus spheroids, so this is actually, we're just comparing cell lines at this point. So differences in gene expression between the cell lines, you can see we have thousands, 5,000, 6,000 genes that are differentially expressed between the cell lines themselves, allowing us to be able to differentiate between a biphasic mesothelioma, an epithelioid mesothelioma, and of course any of the meso versus normal mesothelium and versus lung cancer. So we have a whole lot of potential candidates to look at. We're not going to look at 6,000 genes at one time, I hope. But some of the genes that come out of our studies, we do validate using quantitative real-time PCR. So we take our cells grown as a monolayer, we grow them also as a spheroid, and this time we were able to obtain some fresh frozen tumor sample. So this is a big giant tumor that comes out of a patient, and then this is the tiny little bit of sample that I get to work with. So I extract RNA from all of these three conditions. I do quantitative real-time PCR, and I look for the expression of our potentially interesting genes that come out of our microarray data. And a couple of the genes of interest for us are genes like CCL2 and MMP9, which are involved in the immune response. And when we compared their expression in the cell lines versus fresh frozen tumor, here you can see, so the fresh frozen tumor I've colored in green. We had two different samples. And you can see, especially for MMP9, MSTOs grown as spheroids more accurately mimic the expression that we see from fresh frozen tumor samples. CCL2 came out of the microarray as a potentially interesting gene. It wasn't as high as MMP9, but you can see here that the MSTO cell line is more closely related to what we see in vivo than any of the cells grown as monolayers. So our take-home message: our 3D model appears to more accurately mimic mesothelioma in vivo. We need more fresh frozen tumor samples to continue our comparison, and we need more genes to look at. We do have lots of potentially interesting genes. Unfortunately, we still don't have one target that we have found. And more effort is needed to find gene targets that will allow for earlier detection and hopefully lead to better prognosis for our patients. And with that, I will end and thank my coworkers, my awesome lab, and I'm happy to take any questions.